Thank you all for coming, and thank you to the AKA for letting me present this stuff. I'm going to present this stuff about burnout, and it's quite personal to me because, you know, as Kathy mentioned, I've been travelling a lot over the last almost 20 years and teaching, and I guess Philip is here, and Charles somewhere will testify how stressful that can be. It seems glamorous that we travel, but it isn't, and how many relationships we've got behind us, you know, as well. You can count them on your hand or two. And so it is a big sacrifice that we make by being developers and travelling a lot, and I guess people don't really realise that. Earlier in the year, when I was getting tested by a naturopath, it came back, my cortisol levels were low, and I freaked out because what I realised is I'm heading to burnout. And I've been getting all these ideas about burnout and trauma and PTSD. I had a model in my mind developing of how it is. But what I realised is that I better write this balance because I need it for me. So it was a little bit selfish. And then I gave it to one person who works closely with me the last 10 years, my ex-wife, who's actually knows a lot about the clinical applications of NK, as she's a psychologist and a psychotherapist, and she does a lot, and she works with the work in a psychiatric hospital as well in Austria. And she came back and said to me, Hugo, this is the best balance you've ever written, and they're actually seeing changes in psychiatric patients in the hospital that the doctors can measure on their machines, and they said it's the only treatment that's worked so far. So that's kind of interesting. So, and I'm going to present, and I've put some of the information in your manual you have there is a case study I did with the heart rate variability meter, and which is a very excellent way of measuring this kind of thing. But anyway, so the reptilian brain and burnout is this. Okay, now it's working. So the first part of it is this polyvagal theory. I don't know how many people have heard of this. It's kind of this new thing coming up. Oh, it's not new, but maybe in kinesiology people are starting to talk about it. And it has 3 stages of the vagus nerve. Now, this is not the 3 stages of stress. People often confuse them. It is not. The first stage is the unmyelinated vagus nerve. And the unmyelinated vagus nerve is the very primitive or reptilian vagus nerve is a good name for it because it's present in reptiles, and as we'll see, when it gets overstimulated we start adapting like reptiles, which is not optimal for mammalian survival or adaptation. Unless anyone here has reptilian tendencies, I don't know, or your clients. Or you have a snake or a turtle as a client. Don't laugh. I mean, working on animals, it can be big business. I know many people who have made a lot of money working with kinesiology and animals, so it's not a joke. But you have to remember that. So basically, it actually is good for our digestive system. So normally it fosters the normal digestive process through the vagus nerve, but also it's responds to threat by, they call it immobilization behavior, but it's actually the defeat response. It's not like, oh, I freeze, there's a danger far away, what am I going to do? You know, now it's coming to me, I'm going to run. What it is it's like freeze, belly up. I am defeated. It is the belly up response, so it is actually when you go into the exhaustion phase and 3rd stage of stress, that's part of the contribution to it. So then I realised, okay, that's part of the story. Okay. The second stage is what they call is vagal withdrawal. That means the vagus nerve withdraws and the sympathetic nervous system rises, and that's basically your first stage of stress. The third stage is this myelinated vagus nerve, which is very interesting because it actually regulates your cardiac output, but also your social engagement system. It's actually the no stress state. It's when you're not stressed and you can actually engage socially with people. And I thought, well, that's pretty interesting, because that's actually part of the whole stress model. Now, you're all aware that there's 3 stages of stress. I actually refer to them the 4 states, because I think you have to consider the no stress state as a state, which is the optimal state. And we know that is, you know, like when you're balancing someone with kinesiology and they go into that really relaxed, chilled out phase, that you're feeling really relaxed and, oh, that's so great. Or, you know, like sometimes when people are working in one of my courses and everyone's in this parasympathetic, no stress state, you can feel it in the room. It's really amazing. I'm sure all of you have had that experience where you've got a collective state in the classroom while everyone's practicing. So I just want to quickly go over these physiologically. This is the 3 stages of stress from Hans Sely, the Austro-Hungarian scientist. I went to Hungary once and said he was Austrian. He's apparently, you know, he was born in Hungary but worked in Austria. I don't know, whatever. Anyway, so the no stress state. So this is the myelinated vagus nerve that fosters this social engagement. And what it has, the social engagement, if you look at that, you know, and I think this should be in your manual, it's got like opening your eyelids, looking, listening, speaking, engaging. You know, ever been, you know, especially in a relationship, you get a bit stressed out, you stop engaging, and think, you're not engaging with me. Anyone ever had that experience? Yeah, right, because it's about, you need that to engage. Right? And if you get stressed out, it goes away. You can't. You know, under stress, you can't socially engage with people. So this is very important and part of the myelinated vagus nerve. But it also is very important for the very relaxed state of the innervation of the heart and the lungs, where you go into this really relaxed state. It slows it down. They call it the Vagal Brake, which I'll show you in a minute. So these, this is located, the nucleus ambiguous in the medulla is where the innervation comes from, and it's part of this inhibitory component of the parasympathetic nervous system that communicates with your sinoatrial node in the heart, which basically regulates your heart rate. So it slows it down, okay? But it's calming. It's restorative. It inhibits the sympathetic nervous system. It lowers your blood rate. It lets you breathe easy. Sounds pretty good, right? Well, that's where you want to have your clients, right? Okay. First stage of stress, nothing new, sympathetic nervous system, adrenaline, adrenals, the periaqueductal grey matter of the midbrain. This is your fight-flight. When people are in first stage in stress, how does it look like? Or if they're irritated or start getting anxious. You know, if anyone's a little, you know, sometimes you get mildly irritated. That means, if someone's mildly irritated, it means the sympathetic nervous system's kicked in a bit, and they start losing the ability to socially engage. You go, oh, you're irritated and now you're not engaging with me. You know, and then it starts, you know, then you get more stressed out, you become more unable to socially engage, and the more the other person gets here and it starts going do, 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 do. Anyone ever had that experience? Yeah. Yeah, me too, don't worry. And the other one is anxiety. If you go, when people start getting anxious or start worrying or fretting, that means they've gone into the fear component of the fight-flight, okay? Second stage, this is the HPA axis, you know, the hypothalamus, pituitary, adrenal. Basically, it's cortisol. I mean, people think of cortisol, ooh, bad. No, it's not bad. Cortisol is good in the right amounts. Too low is bad, too high is bad. But the right amounts is good, okay? And so this is interesting when we come later to look at what we're going to talk about with the third stage of stress, when you've got low cortisol, or adrenal fatigue they call it, or adrenal burnout, or whatever, you know. But basically that's this idea of low cortisol, and it's what made me take all the ideas I had and put them on paper and start developing what I had, and sort of tweak it a little bit so it would work with, so we could actually do balancing for this. Third stage of stress: burnout, PTSD, trauma, anxiety disorders, all this kind of thing, you know. Like, it's really a lot of it, because, you know, what happens is when you're in too much of this, then at some point you start burning out. And what was interesting to me is, like, yeah, we used to go, oh yeah, you know, parasympathetic nervous system good, sympathetic nervous system not so good, and long-term stress not good, but they're all like there for a reason. But if you're in long-term stress for too long, that is, your cortisol levels are high for too long, and then you start going into the fatigue, the burnout, or the third stage exhaustion phase, which we saw with that diagram. You know, it goes up there and then down you go. So, so the model that, you know, and this is interesting, is with the thyroid hormone, reverse T3, the unmyelinated vagus nerve, and low cortisol. And it was interesting, about a year and a half ago I was teaching my hormone course, and I've got this whole flow chart with thyroid hormones, and it's got reverse T3 in there, and, you know, because it's made in the liver and blah blah. And someone asked me this question, and it started making me think, well, why the hell do we even have it? I mean, we have an enzyme that makes it. I mean, what's that about? Why? And so that started making me think, well, you know, like, as we look, it actually could contribute to the burnout, or the third stage of stress, because it blocks the action of the real thyroid hormone T3. It stops it from working, so therefore it inhibits your metabolism, which is what you observe when people burn out. So this was my idea, and so, like, earlier in the year when I realised I had low cortisol, I thought I'd better write this balance, you know. So these were the three things that actually were like the model for burnout. Okay, so let's look at the thyroid hormones first. And so with the thyroid, that's produced from tyrosine and iodine. Okay. Now the two products of the thyroid gland are T3 and T4 thyroxine, and T3 is the hormone, it has the receptor. T4 or thyroxine is not a hormone. There is no receptor for T4. It actually has to go to the liver and be converted to T3, and it's a secondary mechanism your body has for regulating T3 levels so we don't have too much. Because if you have too much thyroid, you have hyperthyroid, like Graves' disease, and that can be very serious. Okay, so it's an extra mechanism of modulating the levels of the thyroid hormone. Cool. And this is, I love my pictures. This is a good one, you know. I guess your right brain kinesiologists love pictures too. But basically, iodine comes into the thyroid gland. You've got this protein called thyroglobulin. It's then has iodine molecules put on it. Then the tyrosines are lobbed together like this, and you get either T3 or T4 depending on the number of iodine molecules. The 3 and the 4 just simply relates to the number of iodine, and then they go taken out into the blood either as thyroxine, which is T4, or triiodothyronine, which is T3. Okay, very simple. Now in the liver, what happens to T4? We've got two enzymes, and this is, I like all the technical things, you know, these are the enzyme numbers if you want to search the enzyme database. This is the DIO1 and DIO3. They're the gene codes for the enzymes. If you want to look up the the genetics, the actual about the protein, that's where I go and find the information. Like web pages like Gene Cards or NiceSign for enzymes. And so this one converts up here, the top one, T4 to T3 and also T3 to T2. So it actually breaks down T3 into T2 as well. So it's a way of actually, like everything in the body, what's kind of interesting, we think, normally what do you think about toxicity? You say toxicity is getting toxins out of the body. But the toxicity pathways, do you think our bodies were designed to get rid of pesticides out of a body? No, they weren't. Because when our bodies were evolved or designed or whatever, what happened is there was no toxins or pesticides, but that system is designed to clear normal biochemicals out of your body. So the detoxification pathways are not just about getting rid of toxins and stuff that shouldn't be there. It's also about getting rid of stuff that should be there. And sometimes people forget that, and that's a whole other story, And so we have to clear it out and deactivate T3, get rid of it out of the body, and then we make some more. So we have this constant cycle of renewal in our body. So down here we've got this one, the DIO3 is T4 to reverse T3 and also T3 to T2. T3 to T2, not reverse T3. What does that mean? We don't have an enzyme to even break down reverse T3. We've actually got to get rid of it. When they do all the tests, we actually got to get rid of it out of our body, like just in the excretory pathway. We can't actually break it down and deactivate it until it's cleared from our body. Interesting point, they're called a selenoprotein, where they have this special, like I always thought there was 20 amino acids coded into DNA, but there's not. There's this 21st called selenocysteine, and it's cysteine they take the sulfur off and you put a selenium on it, and there you've got selenocysteine. And so we need selenium for this, and in Australia we have low levels of selenium, right? So if you have selenium issues, you could have thyroid problem issues. So it's something to look at if you've got thyroid patients, or clients I should say, that you might need to balance or whatever, refer to a naturopath for selenium supplementation. What's the difference? Not much. On the left you've got T3 and on the right reverse T3. You can see the only difference is where the iodine molecule's taken off. On T3 it's on the end one, and on reverse T3 it's on the one closest to the carboxylic acid group and the amine group. So actually, technically, if you wanted to, they actually fit the definition of an amino acid, which is kind of interesting. So, but anyway, that's what they are, and so slightly different. But it's a massive, massive issue with, if you take the wrong one off, it totally changes it, because though it'll still bind to the receptor, the reverse T3 blocks the receptor, whereas the T3 activates the receptor. And T3 receptors, this is another interesting thing you may not be aware of, are a class of receptors called steroid receptors. They're found inside the cell, and they activate the DNA expression to produce protein. So that's kind of interesting. So this is how it stimulates metabolism by causing the proteins to be produced you need to metabolize all your other biochemicals. So reverse T3. So I did find one use for it. Interestingly enough, it's this enzyme that produces it. So this is what happens when you investigate the actual protein and the gene. So I investigated it a bit, and it found out it's highly expressed in the pregnant uterus, placenta, fetal and neonatal tissue. So there's a proper role for it. I'm like, wow, that's interesting. And it prevents the actual fetus from being exposed to a adult levels of thyroid hormone. And people who have issues with that gene have a very poor development. Because they have too many thyroid hormones during development. And if you understand embryology and all that, the thyroid hormones are a very important part of development. So, so what happens is that reverse T3 is actually produced under extended periods of long-term stress. That's what they've found. So when you get, obviously you have to get tests done to actually find out levels of reverse T3, and just the general thyroid tests won't cut it, because, the two molecules are so similar that if you weigh them, they weigh the same because they've got the same chemical formula. It's just the arrangement's different. And I always say, anyone who's trained with me knows rule number one of biochemistry: structure governs function. You change the structure of something, you change the way it functions. So you slightly change the structure of T3 and you change the way it functions, and that's the difference between T3 and reverse T3. So theoretically, the T3 could contribute to burnout, because what it does is it actually blocks the T3 receptor and therefore stops any good T3 from getting to it. So it's like the double whammy, I call it. It blocks the receptor, but also the receptor doesn't get activated, but it also, by occupying the receptor, it stops the proper T3 from being able to function. So you get two effects from this. And so this is the whole thing. Well, the jargon in the, say, naturopathic circles is, if you've got too high reverse T3, they would call it reverse T3 dominance, which means that the levels of reverse T3 are too high compared to normal T3. Okay, and so understanding this, I thought, that's got to contribute to it, So I made this, I think you've got this, I put this in your manual. You can see here, this is a summary of this pathway. Your thyroid gland, T3 and T4 are produced. The target tissue has the T3 receptor. The T4 goes to the liver. Here you can either produce T3 or reverse T3 under long-term stress for a long time. You start getting higher levels of reverse T3, and when the reverse T3 dominance seems to coincide with when you start shifting into the third stage of stress, so therefore, it seems to be relevant. And just in terms of the kinesiology and the acupressure formatting that we've been using in NK, certainly we've found that the whole idea of T3, reverse T3 and the T3 receptor in the target tissues has been showing up with bundles of stress when you work with people with trauma or PTSD or burnout or anything like this. It really becomes a massive thing to clear. Okay. Some of the functions of T3, this is what it should be doing. Your lipid, carbohydrate, protein metabolism, hormone production, degradation, but also oxygen consumption. I remember Charles talking about the oxygen, how important lack of oxygen is, and he developed a whole protocol for it. And, really this, T3 is really important for that. So if you've got too high levels of reverse T3, what's going to happen? You're going to have, it's not going to work, you're not going to be able to consume the oxygen. So you go into this hypoxia state. We were talking about that last night, about hypoxia and neurons, how they can all of a sudden be active after 40 years. And he was talking about his somatosensory neurons, and they're working after like almost 40 years. It's like, say what? They were there and all of a sudden now they're working. It's like, and this one of the contributing factors, kind of interesting. So there's basically, you can have two major types of response of thyroid hormones: cellular differentiation and development. So that would be very important in embryo life, but also in tissue that is evolving. tissue that, you know, I mean, not involving, that divides. So something like the gut, where you've got, you have to renew all your gut cells all the time. They're renewing. And so, you know, they die and then they divide. Wherever you've got cell division, you need the thyroid hormone. Of course, if you've got low levels of it, it's going to happen, not much. Okay, lot of effect on behaviour, especially behavioural changes, growth, cardiac output, immune system. I mean, it's really widespread. So it kind of fits that, you know, hypothyroid state can be induced into the human body by producing more reverse T3. Then that fits in with the model of it works in burnout, especially if it starts getting produced under long-term stress, and its effect of cortisol on the liver, high levels of cortisol in the liver for a long period of time. You get the reverse T3 enzyme instead of the not reverse T3 enzyme. And then these are just, you've got this in your manual anyway, so these are just some of the symptoms of hypothyroid, which you know, you start seeing in burnout clients and trauma. So the second sort of point that, or thing that I wanted to look at with it would be the unmyelinated vagus nerve. And so this is kind of, or the reptilian vagus, as we called it in the talk, because it is the reptilian vagus nerve. And so what it does, it's actually associated with immobilization behaviours. And what they mean by that means like the defeat, the belly up, the feigning death, you know, passive avoidance, behavioural shutdown, syncope, which is fainting, fancy way of saying fainting. It reduces the cardiac output through innervation of the vagus nerve, the innervation of the sinoatrial node in the heart, and it also reduces oxygenation of the blood in the lungs as well. That's pretty cool if you're a reptile, actually. You notice how they can do that? Yeah. So with the cardiac rate, I just want to speak a little bit about that, because I just want to show you this case study I did with this balance, and literally 45-minute tests between before and after, and I just did this balance on the heart in between. So the myelinated vagus nerve, and it inhibits the sympathetic nervous system. Well, they work against each other. It's a parasympathetic, sympathetic dance. You know, they're always, and especially in the heart. I mean, if you didn't have sympathetic innervation of the heart, you would have troubles. If you're lying down, you know, when you jump up all of a sudden, your blood pressure has to raise and your heartbeat just goes boom, 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 boom. That's the sympathetic nervous system. And that's not first stage stress, that's just the sympathetic nervous system doing its job to speed up your heart when you need it, because you stood up all of a sudden. But then, if you rest, your heart has to slow down. So you've got the vagal brake, and this is the myelinated vagus nerve. They call it the vagal brake. It just slows it down. It goes, whoosh, it slows down. And so you get this variation in heart rate. And the measurement of heart rate variability is a very accurate predictor of actual mortality. You know, and so we started doing some work with some medical devices that measure it. Okay, and so this is the this also raises and level the tone, the cardiac tone, which basically means the number of heartbeats per minute. If your tone is too high, you have excess sympathetic nervous system, that's actually really dangerous. So you actually want to have a lower heart rate. You know, if you're beating at 80 beats per minute, you're like, you know. We'll see, I'll show you that in the test, it's quite interesting. So when the parasympathetic innervation of the heart has problems, the other systems have to kick in. So this is where you get the sympathetic nervous system, the HPA or the unmyelinated vagus nerve, depending on what level of stress you are, they kick in and start, you know, doing what, you know, trying to compensate for the lack of the myelinated vagus stimulation of the heart. Okay? So it might be, it's adaptive in the short term, but in the long term it's very damaging. So this is the whole stress adaptation system that we have. Okay. So the unmyelinated vagus nerve, it comes from the dorsal vagal nucleus, not the nucleus ambiguus, so very clear distinction there. It provides a low tonic influence on the heart and the bronchi, and this is because this is what the reptiles had, and this is what they needed. The reptiles, you know, they can go into a very low heart rate and very low oxygen consumption rate. Being half Ecuadorian, like I've been to the Galapagos Islands many times, and watching the marine iguanas is quite fascinating because they can shut it all down, dive all the way down for all this time, and munch up all their seaweed, and then come back and then sit on their rocks and get their sun. The same with turtles or any other reptile that actually lives in the water, you know, especially in the ocean. Sea turtles and the marine iguana and the snakes as well that we've got up here. Kind of interesting. But mammals have a greater demand for oxygen than they do, and this makes us vulnerable. And so this inhibitory input from the unmyelinated vagus nerve into the sinoatrial node of the heart actually creates this hypoxic state, because it slows down the heart rate, but also it goes into the bronchi and the lungs and slows down the oxygen absorption from the air. So not only have you got less heart arriving at the lungs, we've got less oxygenation of the blood. So that's great for reptiles, but it's bad for mammals. So when we're in this state as humans, this has to be addressed. So it's involved with bradycardia, which is a low heart rate. The tachy one is when you've got high heart rate, you know, tachycardia. That's how I try to remember it, you know. Anyway. So it's important to note it has normal functions. So under normal conditions, it provides a tone to the gut and promotes digestion. But what's interesting, when it gets overstimulated when you go into burnout, it actually causes stomach ulcers from gastric secretion and colitis. So, you know, if you've got clients with these conditions, you might want to start thinking, maybe we should start investigating burnout. Maybe we should check their reverse T3. You might need to start looking at it as a global perspective. Neurochemistry, because I like neurochemistry, and you know, everyone knows me and all my formats, you know. But anyway, preganglionic acetylcholine. The difference is in the myelinated postganglionic, we've got acetylcholine. In the unmyelinated, and this was interesting, different neurotransmitters, and this has proved interesting in far as the balancing goes, just having this knowledge that we can use vasoactive intestinal polypeptide and nitrous oxide as neurotransmitters for the postganglionic, and we start looking at these receptors, especially the VIP receptors in the target tissue, along with the reverse T3. Cortisol. So cortisol, this is your daily what's supposed to be your normal rhythm of cortisol. So often if you're getting a good test, you can do the 24-hour, I did this 24-hour salivary test where I had to spit in a tube every 6 months. That was a 24-year test, you know. I'll finally get the results after I died, so anyway. Then they'll work out I'm really not that stressed out anymore. I'm in peace now. Anyway, but so what happens, this is the normal daily rhythm. And so with the low, I think I had 3 out of 4 low, but overall it was low, something like that from memory. So kinesiology, balance cortisol production in the adrenal cortex. Okay, so very first thing, balance the cortisol production in the adrenal cortex. We need to look at the reverse T3 receptors and the unmyelinated vagus nerve. Just that alone, in what we've found is, because the balance I've got is quite long, just doing that is pretty powerful, and it's enough. And then we come back later and do that. Then we balance heart, lungs and other vagal targets, especially the heart and lungs, because of the low cardiac output and the lack of oxygenation of the blood. And so this will certainly address any hypoxia that's going on, and people under long-term stress or have been stressed for a long time should have this looked at. And then we could balance the sacral parasympathetic nervous system as well, because that gets the last part of the the colon, but also the reproductive glands, the uterus and all this, conceiving or any stress there. So actually looking at that component of because we generally think of the parasympathetic nervous system and the vagus nerve as the same thing, but they're not. The vagus nerve is only a part of it. There's lots of other nerves that contribute to the parasympathetic nervous system. Okay. Just a tip, time of day balance. Well, we all know that, right? Everyone should know that. I've been using it a lot in jet lag balancing and stuff like this. So I had the idea, because of this cortisol rhythm, to use this. And this is just from Chinese medicine, the activation, but you can use it. You can use the alarm points as a time of day, and use it, add time mode if you want. And then do the alarm points with time mode. And what we've found is that often we get this stomach between 7 and 9 in the morning showing that's when your cortisol should be peaking. Except for one person who had this issue, and it was showing up 2 in the afternoon, and we go, what's that? Well, this colleague of ours in Austria, she works as a kinesiologist, she works in our institute there, but she also is a marketing manager of Porsche in Salzburg part-time, and at 2 o'clock every day of the week, usually, that's when they have their team meetings. And so that was pretty interesting. You'd imagine being a marketing manager of Porsche, automotive company in Austria. I mean, it's a pretty full-on job, But anyway, so this would be the thing there, and so don't forget that. I find it very useful. And for those of you who use acupressure formatting, you can just put physiology mode and test the alarm points and use it as the time of day, or with time mode as a time physiology to the previous format that you've gone on. Okay. So the case study, I've got like 5 minutes left, I can show you this. This was done on a 76-year-old male. He was 173 metres high, 73 kilograms, so not... Huh?
You said 173 metres, that's pretty short.
Oh, okay, whatever. 173 centimetres. I did the 2 tests 38 minutes apart, and I did the unmyelinated vagus nerve on the cardiac ganglion in between, and the testing was done with this biosign. This is from Munich in Germany, a heart rate variability scanner, proper medical device. And I even sent the results to the guy and said, he helped me fix the data up. There was a couple of artifacts we had to get rid of, and then he was very impressed with it, But it's just a one-off case study, so it's just a case study. Understand that, okay? So there's various tests you could do. We did the deep breathing test, and that tests the current state of the parasympathetic nervous system, which is responsible for relaxation and regeneration. So this is basically testing your vagal braking, which is how well your heart, the parasympathetic nervous system can slow down your heart. There's 3 testing things that they had: flexibility, tone and dynamics. Flexibility represents, basically it's the measure between the top heart rate and the bottom heart rate. This is a measure of the heart rate. So if it's very close, it's not very, very flexible, that's bad. It should be very flexible, okay? This is the dynamics. This is the gradient of how fast it slows down. A good one should slow down really quick. That means your brakes work. That's your parasympathetic brake. So it means you slam on your brakes. All of a sudden someone stops in front of you. You want to have good brakes. If you've got crap brakes like this one, you're smashing into the guy. So that's dynamics. And then the tone is just your average heart rate. Should be around 60 or something. Up here, 100 bad. This is good. So this was before. The percentages are the percentage for his age group. So he was 67. So he was healthier than 20% of all other males at 67 years of age. It's not so good, is it? No. And then after, look at the difference. And you can see, like in the dynamics, he almost went up to 100%. Tone up to 60, flexibility 60. I mean, that's after 38 minutes. And the changes, you can see, this is the gross change in percentage for his age. So he went from 20% to 65, and, you know, here around 20 up to 85, and, you know, up to 55, like this. So this was the before. That was his measurement. And this is the after. And you can see they've got this is the change from the previous. So the actual raw data. He was ranked 18.6 and now he's ranked 63.8. So they're basically percentiles. And these are the percentiles for his age, you know, 5, 50 and 95%, like that. So 95%, if you're healthier than 95%, you'd have a value of 22. If you're healthier than 50%, you'd have a value of 14, and 5%, you'd have a value of 5. So, and then you've got here some, just some of the data and the percentage change. And I was interested to see the percentage change. I put some of that, I think, in there. And so you can see here that some of the percentage change were really high. You know, the tone went down. You want a negative. Some of them you want negative. But look at his biological age. Before he was 74, now he's 58. I mean, that was not bad. He's 67. And, you know, so, and, yeah. So that's it from me. So thank you. And I've got a few minutes for I know some people shot their hands up, so anyone got questions? Yeah, Elizabeth? No, I took the readings directly after the balance. I did the readings. 38 minutes was how long it took me to do the balance, and then I did it again. I haven't retested him, if that's what you... Yeah. Richard?
RT3 in New Zealand, you can get TSH, T3, T4, but you can't get a T3 reverse T3 test.
You can't. In New Zealand?
They don't have the labs in New Zealand to do it.
They send it here.
They send it to Australia?
Yeah.
At a cost.
Louise is from New Zealand but lives here and is a naturopath, so maybe you could talk to Louise. Okay, anyone else? Okay, thank you.